For those of you who don't know me, my name is John Rex, and it's my honor to open up the boot camp this afternoon. And so thank you all very much for being here. Actually, I'd like to thank a couple of folks real quick before I forget. The groups that have supported are listed on the slide. It's all of the sort of a collection of funders and interested parties who work closely together to bring things to, like this to be. As you'll see in the course of the afternoon, there'll be coffee and cookies, and then there's a stand-up dinner buffet after that, and all that's being provided by this collection of folks. The program committee has kindly allowed us to begin to add this on a regular basis to the overall meeting. It remains something of an experiment. We still think it works pretty well in this format. And the whole meeting was originally started, as you know, by Hope and Rodval, but this year's chairs are Hope and Sujata Bhavnani. And I think they've done a nice job with the regular program. And thank you all very much for being here this afternoon. I know it's hard to get here. This is the Tuesday after Labor Day in the United States. I get it. The flow of the afternoon is in your program. Basically, it's two-hour blocks with a coffee break in between. We will be done by 5:30, at which time there'll be another event. During the afternoon, as you listen and think of a question, you'll find index cards on your table. Look around, find them, they're there. Write your question down. There are several of us who will be circulating the room and collecting index cards. If you write down a question and give it to us, we'll give you back a blank card, and you can send in another question. And we promise to anonymize the questions, so feel free to ask your most pointy, difficult question. On your way in, very importantly, you received a colored wristband. I hope everybody has one. If you don't have one, you can get one at one of the breaks. During the reception that follows, and by the way, the coffee and the reception will be through the doors that are in the back of the room. You may not realize it, but that opens up. There's a whole other room on the other side, and we'll have all this space available for you to move around. But the food and the good stuff is be on the other side of the doors. We're going to have a 400-person, actually 450-person, if everybody turns up that's registered, speed dating game afterwards. We will, at periodic intervals, ring a bell and encourage you to find another person with the same colored name tag and same colored wristband and introduce yourself. I'll remind you again of the rules of the game. It's meant to break down that social barrier, and I want you, by the end of tonight, to have made at least a few new friends. Yeah, I'd have no idea whether there'll be an opportunity to collaborate, but you won't know if you don't meet them, and so I guarantee you this actually ups your odds a little bit. We are videotaping this session. It will be all replayed live ultimately on the web, and so that's why the front of the room is where the questions are and where the panel is. And I think I have said everything I need to say, and so I am now going to turn it over to Ursula.
Welcome all to the first bootcamp of this conference that will focus on the emergence of mutational resistance and predictive models in the discovery and early development phase. It's a very complex issue with lots of challenging questions, and we have 3 speakers today with extensive experience with many different programs, and they will have different approaches, different views, and different perspectives on this topic. You will see it's not possible to give recipes to solve this complex topic, but we will try to give you some perspectives and maybe inspire some thoughts how to deal with this topic. 2 examples illustrate the complexity of this topic. 2 antibiotics that are single target antibiotics. I chose that just as an introduction to this workshop. It's mesillinam and the JSK boron compound. They have certain characteristics in common. One is they have both quite a high frequency of mutation. They have large mutational targets, and they were both used for the treatment of urinary tract infections. If you would have stopped here, you wouldn't assume that there were different clinical impacts with these different antibiotics. In case of mesillinam, the laboratory mutants did not predict the clinical mutants. These clinical mutants were not target-based. In case of the JSK compound, the laboratory mutants predicted the clinical mutants. And they were target-based, mainly target-based. For mesillinam, the mutants were fit, but they did not maintain resistance in the urine, whereas for the JSK compound, they were fit and they supported maintenance in urine. So in case of mesillinam, the mutants in the clinical situation in urine were not resistant, and the JSK compound, they were fit, stable, and all fine. The clinical impact for mesillinam is that it's still used as a useful antibiotic for the treatment of urinary tract infections, and the JSK compound failed in phase 2 clinical development due to emergence of resistance in this clinical study. So this illustrates how important it is not to stop at very simple assays and just assume that the frequency of mutation is enough to predict the clinical impact.